Good afternoon. Thanks for joining us today. We'll give it a few minutes for other attendees to hop on before we get started. Afternoon. Thanks for those of you who are signing on. We're just going to give it another moment or two for others to join us. All right. We're a couple of minutes after the hour now, and I think attendance has slowed down. So I think we can go ahead and hop in. My name is Jules Savat. I'm a genetic counselor by training at Geisinger Health System in Pennsylvania, where I help facilitate GenomeConnect, which is a project that's part of the Clinical Genome Resource, an NIH-funded effort. And I am really excited today in collaboration with NothingPink. We are hosting this lunch and learn to share a little bit more about genetic information and data sharing. So thanks for taking time out of your day, joining us, and we're excited to have you. Carrie from NothingPink, I see that you're on. I'm not sure if you wanted to say anything else in introduction before I hop in. If not, I'm happy to jump right in, but wanted to give you space in case that's helpful.
Sure, wonderful. Thanks so much. We are thrilled to have this resource to be able to share more education about genetic testing and what our information can do when shared. So we have a number of people on the call and a number of folks really interested in learning more about this. So thanks so much for doing the webinar with us today.
Thanks, Carrie. So I'll hop in. Really excited to have you all. To give you a sense of what we'll talk about today, I have slides to walk through and we'll have some time for a question and answer at the end. A couple of other names that you'll see on the call today are other GenomeConnect team members, so they might be managing the chat, Caitlin, Cooney, and Julia. If you have questions during the webinar, you're welcome to put that in the Q&A box. You won't be able to unmute or come on camera, but we're happy to get to those questions once we have some time at the end of our chat. We do ask that because this is a recorded presentation, you should see a little banner at the top that this is being recorded, that as you're asking questions, to not share personal medical information, but we're happy to answer kind of broad questions about genetics, about data sharing in our discussion today. So what I will talk about before that Q&A time is a little bit about genetic data. What is genetic information, kind of level setting? Many of you might be aware of genetic information given your own genetic testing journeys, but we'll talk a little bit about that. And then we'll move into how individuals can participate in sharing their genetic information. And as Carrie alluded to, the power of that sharing. So why is that data sharing important and how can it benefit the broader community as well as individuals engaged in that data sharing? So to get started, let's begin with what genetic data are and how we interpret genetic information. So each one of us has more than 1 trillion cells that make up our bodies. We have different types of cells, hair cells, skin cells, brain cells, and they're our building blocks. In each of those cells, all of our genetic material or our DNA is wound up really tightly. Our DNA is like our body's instruction manual. Each instruction is called a gene, and each gene codes for a protein. Those proteins have different instruction or different roles in our body. Some help us prevent cancer. Some help us digest our lunch. Some are part of how our brain develops and grows. So each gene gives an instruction for that protein, and then those proteins have different roles in the body. Our DNA provides instructions through the letters A, T, G, and C. So our DNA is made up of nucleotides that are those letters. Our body reads those nucleotides or letters to then make those proteins. We all have changes in our DNA in those letter sequence. And some of those changes, or what we call variants, are part of what make us who we are. But some of those variants can affect how our body reads those genes, how those proteins work, and can lead to developmental or health differences, or can increase someone's risk for cancer. About 5 to 10% of cancers are thought to be caused by genetic variants that a person is born with. So a change in that letter sequence of the DNA that impacts how a protein works in the body and puts that person at a higher risk for cancer, put them at a higher risk for cancer. There are certain personal or family medical histories that increase our suspicion that there might be something genetic going on. One, if someone in the family has had genetic testing and such a variant was found. If there's a certain pattern of cancers in the family, like cancer being diagnosed at an early age, often before the age of 50. If one person has had several different types of cancer, so maybe a breast cancer and an ovarian cancer. If cancer was diagnosed in multiple family members, in particular, if it's the same cancer or a certain group of cancers, maybe a mom and an aunt on that side of the family, both with a breast cancer. If someone has cancer in both organs, so both breasts, for instance, or both kidneys, if it's a paired organ, certain rare types of cancer, like a male breast cancer, and then also being from an ancestral group that's known to have an increased risk of having certain genetic variants. For example, individuals with Ashkenazi Jewish ancestry have a higher chance of having some of these genetic changes that increase the risk for cancer. If you have questions about your personal or family history, we encourage you to talk with your doctor or genetic counselor. And we have a link where you can find a genetic counselor in your area, which one of my lovely colleagues will put in the chat as we're talking today. But all in all, these genetic changes that increase the risk for cancer can be identified through genetic testing. And there are many different types of genetic tests. And some of you might be familiar with one or more of these tests through your own genetics journey, through the own testing that you've had done. Typically, these tests are done using blood or saliva samples, and the different tests look for different types of genetic variants. So we have a couple of genetic tests, a karyotype and a microarray, that look for large missing or extra pieces of genetic material. And then we have sequencing tests that actually read through each individual letter of our DNA, looking for changes in those A's, T's, G's, and C's. So really different tests to look for different types of genetic changes. Often in individuals who have a concerning personal or family history of cancer, sequencing tests are often the ones that we do. And we sometimes can sequence just one gene. We might sequence a set of genes known as a gene panel, or we might look across all of our genes that provide our body with instructions. Those genetic tests are looking for different types of genetic variants, and there are different types of genetic variants. So some are large missing or extra pieces, or some are a single letter change in just one gene. Over the years, our ability to detect genetic changes has really improved with advances in that testing. Years ago, all we could do is a karyotype, and not even recently, we could only sequence one gene at a time. But now we do have the ability to look at lots of genes at once to look for changes in someone's DNA sequence. While our ability to detect variants has really improved over the years, sometimes it can still be like looking for a needle in a haystack when it comes to determining what genetic changes impact someone's health, put them at a higher risk for cancer, explain their cancer history. Sometimes it can be really hard to determine what variants are tied to health and disease and what variants are just part of what make us who we are, aren't going to impact our own health and healthcare. Anytime a genetic testing lab does a genetic test and finds a variant, there are a couple of steps and a couple of questions that they then ask themselves. So they want to classify that genetic variant that they found. And the first question is around, is this gene tied to disease? What do we know about this gene? For many people who are having cancer or having testing related to their personal or family history of cancer, we're looking at genes that we know have a clear tie to cancer risk. If that gene has a tie to cancer risk, the next step is, well, what about this specific change to this gene, the specific variant? Does that variant impact how this gene works in the body? And the lab will ask several questions to try and classify that specific variant. And ultimately, they want to put it into a variant classification category. There are 5 different variant classification categories. Benign and likely benign mean that based on the information available, we don't feel like this variant is going to impact how the gene works in the body, isn't tied to an increased risk for cancer, doesn't provide an explanation for what's going on in the family. Likely pathogenic and pathogenic on the other end of the spectrum, those are genetic changes that, based on the information we do think, are going to impact how the gene works in the body, how that protein works in the body. And then uncertain means we just don't have enough information. Maybe we haven't seen that genetic change in other individuals before, or there's just not enough information to help us put it in a clearer category. The questions that the lab might ask to get to one of these categories include things like, has the variant been seen in the general population? Has the variant been seen in individuals who have a strong family history of cancer or themselves have a history of cancer? Have we seen similar genetic changes? Has anyone studied this genetic change in the laboratory? Was the variant passed through the family and is it tracking with cancers in the family? Were there other genetic changes found through testing? And how does that specific variant impact the protein? And they put all that information together to reach one of those classifications. Now, over time, our understanding of variants can shift and our classifications might shift as well. And we'll talk a little bit more about that later today. In addition to questions around, is this gene tied to disease? Is this variant pathogenic? Is it going to impact how the gene works in the body? Even if we know, okay, this gene, it increases the risk for cancer if someone has a pathogenic variant in it, sometimes we still have questions about that gene and about the condition. For instance, we might be interested in answering questions around, well, do certain variants in this gene cause certain symptoms more often or certain risks more often? So maybe not all of the genetic changes in the gene are equal in terms of the symptoms and risks that we're seeing. What are the frequency of symptoms or of cancers, and when are we often seeing them? And how do we best care for people with changes in this gene that are pathogenic? What additional screenings might we recommend, or next steps for their care might we recommend? So even if we understand a gene really well and a specific variant, we've classified it into pathogenic or likely pathogenic, those categories that say we do think this is going to impact how the gene works in the body, we still might have questions around these sorts of things. And for these reasons, the Clinical Genome Resource Project came about. So this is the NIH-funded resource that myself and others on the call are a part of. And it's a project that's working to build a resource that defines the relevance of genes and genetic variants for use in clinical care and in research. Ultimately, that NIH-funded project hopes to better understand what genes are tied to disease, what variants are pathogenic, and how we best care for patients with changes in these genes. ClinGen does this work first by encouraging data sharing. And this graphic that summarizes the ClinGen project has lots of different pieces on it that we won't get into today, but all of the work within the ClinGen project rely on us collecting as much information as we can, pulling our resources together so that we can answer these questions around what genes are tied to disease, what variants are pathogenic, and how best to care for folks. We're going to focus today on patients, individuals, and how valuable they can be in collecting information. ClinGen has encouraged various community members like labs, researchers, and clinicians to share data, but we felt that patients also were an important source of information because they're often experts about their own health history, can provide more detail about their health, their symptoms over time, and access to those additional details can be really valuable. 10 years ago, ClinGen launched its own patient registry called GenomeConnect as a way to engage patients in sharing their genetic and health information. So let's talk a little bit about GenomeConnect and how individuals can participate in that data sharing. And then we'll talk more about the potential benefits and how that can be useful. GenomeConnect, that patient registry that ClinGen launched back in 2014, is open to anyone who's had genetic testing, regardless of diagnosis or genetic test results. The registry is online, is currently available in English, and we're updating our Spanish documents right now, and those will be available soon. Individuals who are interested in sharing their data sign up and consent on our HIPAA-compliant platform. And in that sign up and consent, they are agreeing to sharing their genetic and health information, and for that information to be shared in a de-identified way with genetic databases. They upload a copy of their genetic testing results, and that's how we pull out the genetic information. And then they provide their health history through surveys. So a first survey collects information about them from head to toe, general information about their health, and then more specific questions are assigned based on any answers anything that they told us in that initial survey. So, for example, that first survey will ask broadly about heart issues, GI issues, cancer history, and then more specific surveys are assigned to help us get more information, drill down over time. This sounds like a lot of work. I'm happy to say that this takes less than 15 minutes for most participants to complete because we're really only asking about the very specific pieces that you tell us about, that individuals tell us about. Participants, as I said, upload a copy of their genetic testing report. They upload either a PDF or image of their report. If they no longer have a copy of their report, they're also able to give permission for our team to request records from the lab that performed their testing or the doctor, genetic counselor that ordered it. Once we have a report on hand, our genetic counseling team, a genetic counseling assistant team, review that report and pull out the specific information about the variant. So what's the gene name? What is that specific DNA change? How is it classified by the laboratory? All of that is pulled out so we have it in a structured way. From there, once we have that genetic information from a participant's report, as well as the health information from their survey, we de-identify that and share it with databases, including the ClinVar database. ClinVar is a publicly accessible database that holds de-identified information about genetic variants and their relationship to health. It's a major resource for labs, doctors, genetic counselors, researchers who are caring for patients, performing testing, and researching genetic conditions. It's a database that's public, freely available for use by those groups, and it is submission driven, meaning that it relies on all of these people sharing data. And it's really how we pool our resources and knowledge in an effort to better understand genetic variants, their relationship to health and disease. So this is what a record in ClinVar looks like. We have additional webinars that we've done in the past if you have more questions about ClinVar, how to search. But if I search a variant, in this case, a BRCA2 variant, the top of the screen will have information about that variant. So it has the variant name up here in the dark blue bar, and then additional information about how we might name that variant here in this box, and that classification or overall classification from all of the submitters. In this case, this is an uncertain significance variant, meaning that all of the folks who have submitted this to ClinVar, there just wasn't enough information to put it in a more concrete category in terms of its relationship to cancer risk, cancer history. If we scroll down, we can then see all those people who submitted the variant. So there are a number first of genetic testing laboratories who've submitted that variant, classified it as uncertain significance. And then you can see our GenomeConnect submission. They're usually at the bottom. We submit to ClinVar in a de-identified manner. So this means that participants' name, date of birth, email, identifiable information are never included. And we're submitting information as it appears on the genetic testing laboratory. So we don't provide our own classification, but if you click in to see more details about what we submit, we do provide how was that variant classified by the lab who reported it, what was the lab who reported it to our participant, and what was the date of that lab report. The real benefit of submissions from GenomeConnect is that we provide additional information about the participant, things like the age of testing, their sex assigned at birth, and the symptoms that they had that they told us about through our health surveys. This is unique to how we submit to ClinVar. Many labs who are submit data in large amounts all at once, and they might not include these specific details. These specific details, as we'll talk about in a few slides, can be really valuable in just increasing our understanding of genetic variants over time. Which takes me to, why is data sharing important? What can it do for the broader community and for individuals who are engaged in data sharing? So first up, we see that in about 30% of our submissions from GenomeConnect, we're the first submitter to ClinVar. Unlike that variant that we just looked at that had many submissions from other labs, about 30%, we're the only submitter in there. And so because the participant took the time to share their report with GenomeConnect, that's how that variant was first put into the database, which means that it is there the next time it's seen by another submitter, or the next time a laboratory identifies that variant in a different patient. So it can be really valuable in increasing the genetic variants in this database. As we just saw, in addition to providing variant information, we provide additional details about inheritance of the variant, that health history. And that can be really valuable in increasing our understanding of what variants and genes cause disease. To help illustrate this, I wanted to share an example from our data sharing. So this isn't a participant who had cancer genetic testing, but I think it's a nice example because it highlights the power of families sharing their information. This is a GenomeConnect participant that I've de-identified for our discussion today, but I'll call them Ella so that they have a name as we're talking through it. Ella was a pediatric patient who had a history of seizures beginning at a very young age when she was just an infant. And she had a genetic test called exome sequencing, which looked at all of her coding portions of her genes at one time. Through that test, a likely pathogenic variant in the SCN8A gene was found. This is a gene that's tied to a seizure condition called infantile epileptic encephalopathy. Her parents were tested as part of her testing, and neither of them had the genetic change, which means that this genetic change was new and unique to Ella, what we sometimes call de novo. Because the genetic change wasn't seen in healthy individuals, was found to be de novo in Ella, and how it was impacting the protein, the lab classified it as likely pathogenic, which, as a reminder, means that they felt like this variant would impact how the gene works in the body, explained her clinical history and that seizures that she'd been experiencing. So although Ella and her family had an answer for her seizures, they chose to participate in GenomeConnect. They enrolled, provided their health history via surveys, uploaded a copy of her report so we could collect those details. We then submitted Ella's de-identified genetic and health information to ClinVar. And at the time of our submission, there were two other records in the ClinVar database. So the first was from Ella's testing laboratory, Lab 1. And their submission likely occurred after they identified this variant in Ella. But a couple of things were notable. They didn't include the health history information, so information about Ella's seizures, and they also didn't include information about the inheritance of the variant. And again, that's not uncommon for labs because of how they're submitting to the database, large amounts of data all at once, that they don't include all of those additional details. Lab 2 also had submitted the variant to ClinVar. And at the time that they had classified it as uncertain, or a VUS, that category where we don't have enough information to say if this variant is tied to health and disease. After our submission, Lab 2 reached out to our team to ask more about our GenomeConnect participant. They wanted to confirm the health history information that they saw in our ClinVar submission and that de novo inheritance. Clarifying that information, confirming that information, then allowed Lab 2 to update their classification. In particular, they weren't able to test parents when they did testing for their patient just because of the situation for the family. But knowing that the variant was new and unique to Ella was enough for them to go back and say, we agree this variant is likely pathogenic. We do think this impacts how the gene works in the body, is tied to seizures. This then prompted them to reach back out to the family that had testing at their laboratory and update their report. So Ella, our participant, and her family, their care wasn't impacted by her family taking the time to share her genetic information or share her health history, but ultimately them doing so allowed another family to get clear genetic test results. And I think really highlights the power of patients sharing their information and having that information be shared with ClinVar for others to use as they're classifying variants as other patients are going through testing. So all in all, data sharing ultimately can be helpful for the broader community because it can help us increase our understanding of genetic changes, their relationship to health and disease. It also can help us answer some of those other questions that I talked about earlier today, like how often are we seeing symptoms? How do we best care for patients? So it can be really useful kind of across the genetic testing and care spectrum. And our hope is that the more information that we collect, the better we'll understand genes, variants, how to care best for patients, and ultimately that can be implemented for patients, for families. In addition to benefiting our broader understanding, individuals sharing their health and genetic data also can benefit them directly. So I want to talk about some of those things before we open it up for questions. First, GenomeConnect can enable connections with researchers or clinicians interested in a particular gene or variant. So it almost works like a matchmaking service to help connect patients, individuals, with others who might be interested in their gene, in their variant. An example of this, if we have a ClinVar submission out there and a researcher reaches out to us asking us about that submission because they have an additional study that a participant could participate in, we share that information back with the participant. So this case is a participant who had genetic testing due to their history of intellectual disability, developmental history, and a variant in the GRIA1 gene was found. They enrolled in GenomeConnect. We shared their information with ClinVar. And after that submission, a researcher reached out to us, letting us know they were working on a research study related to this gene. We then reached back out to the family, told them about that research study, and gave them the contact information for that study if they were interested in enrolling. So it was a way for the family to learn about additional research that might apply to them, and can be another benefit back from the registry. Through the registry, we also can facilitate genetic result updates. We know that families, individuals often want to learn about updates about their results when they happen, but there's not a real consensus in the genetics community about how best to do this. There are often barriers to labs and clinicians recontacting individuals over time. Families might move, their contact information may change, and it can make it really difficult if a lab identifies that our classification of a variant has changed to get that information back to individuals and families. And we realized through GenomeConnect that we could be another way for families to stay up to date about their genetic test results. So in particular, we might, through our data sharing, identify instances when the report that the participant shared is out of date compared to our current understanding. For instance, maybe a family uploads an uncertain genetic variant on their report. But when we go to submit to ClinVar, we see that variant's now classified as likely pathogenic by their laboratory. So this is information that we give participants the option to hear from us, and that we share back if they choose to receive it. If they choose to receive updates from us, we act just like an alert flag for them and let them know we may have identified an update to your result. If you haven't received an updated report, contact your healthcare provider, get back in touch with them to learn more. To show a couple of examples of how this has worked, wanted to share a couple of additional participant stories. So first up, I will call this participant Sarah. And Sarah was an adult with a history of seizures who had genetic testing a number of years ago, and an uncertain result in a gene called CSNK2B was found. This was uncertain at the time. We didn't know a lot about the gene or about the specific variant, but the family enrolled in the registry because they were interested in sharing their information. They were motivated to learn more if they could. We shared this information, shared this de-identified information after we received it, and then became aware that this variant classification had shifted from uncertain to pathogenic. We recontacted the reporting laboratory to confirm that this was correct, and then reached out to the family to let them know there appears to be updated information related to their results, and to reach back out to their healthcare provider. This family, and giving them that update, it clarified Sarah's history, clarified that this was an explanation for her diagnosis, ending that sort of diagnostic odyssey uncertainty for the family. We also have had other cases where we've provided updates that resolved uncertainty by downgrading a variant. So in this example, this participant, I'll call her Beth, underwent cancer genetic testing many years ago due to a personal and family history of cancer. And an uncertain variant in a cancer gene, ATM, was found. Again, this individual enrolled in GenomeConnect. We shared their data with ClinVar. And more recently, through that ClinVar submission, we learned that this variant of uncertain significance, uncertain variant, had been downgraded to likely benign, meaning that the lab doesn't feel like this particular variant would impact how the gene works in the body. We recontacted Beth, prompting them to reach out to their genetic counselor, reach out to their doctor, and hopefully resolve this uncertainty and just provide a bit more clarity about their results. So in some cases, our updating process can provide answers, like in Sarah's case, but in others, it might tell a family or an individual about a downgrade that resolves uncertainty, but and kind of closes the loop on that variant. We often highlight that we return both of these types of updates because I think the benefit is really clear about it provides answers. It helps better understand a diagnosis or a history. But these downgrades, they often are not prioritized by labs in terms of the types of updates that they're sending back to doctors, genetic counselors, and patients, just due to current constraints around time, resource availability. But they still can be valuable to individuals and families because they can resolve uncertainty or perhaps lead to additional testing, additional exploration for them. To date, we've shared over 412 updates with our participants, some that might impact medical care, like a shift from uncertain to pathogenic, others that are unlikely to impact care, but still may be useful for families to learn about. So another benefit back to patients' families who are interested in sharing data is that they have the option to receive these updates from us if we become aware of them through our data sharing. So I know we went through a whirlwind of information and happy to open it up for some questions. But in summary, about 5 to 10% of cancers are thought to be caused by genetic changes that a person is born with that increase their risk for cancer. Genetic testing can identify genetic variants, but questions might still remain about a variant's relationship to health. And even if we understand that variant, we might have questions about the condition. How do we best care for patients with variants in that gene? GenomeConnect and other patient registries, they offer a structured way to share genetic and health information, and that information can be really valuable in a couple of ways. It first can be valuable because those individuals are providing additional genetic and health details, and that can help us answer questions about variants, about genetic conditions. And there also can be some benefits back to individuals. They can learn about additional research studies, and they also can learn about updates to their genetic test results if we learn about them. So with that, there are a couple of resources that I think Julia or Caitlin will put in the chat. If you have questions after today about your own healthcare, we encourage you again to reach out to your doctor or genetic counselor. That Find a Genetic Counselor tool can be a useful resource for finding a genetic counselor in your area. If you have more questions today about, well, how do we name a variant? Or I just looked at my report, what does the word missense mean? We have additional GenomeConnect webinars and videos on our YouTube playlist that go through some of those items, can be a resource to you. And then if you have general questions or just want to read more about the genetics of cancer, cancer genetic testing, there's a useful resource from the National Cancer Institute that I wanted to share. And it provides some overview information that may be useful. So with that, I'll acknowledge that I am the one chatting with you today, and I'm really excited that I had the chance to talk with you. But all of this work is done by a really fantastic team of genetic counselors, genetic counseling assistants, and others, some of whom are also on our call today. Also, if you have questions after today about GenomeConnect or things that we've talked about, you're welcome to email us. The email's there on the screen, and we can drop that in the chat as well. So with that, I'll stop sharing. So in hopes of seeing the Q&A box. And welcome any questions. Carrie, if you have questions or things that you'd like me to talk about more, happy to answer them.
Sure. Well, thank you. I wanted to just express gratitude to all of the team at GenomeConnect and the work that you're doing and how important this is. I think highlighting, that our information can help others is certainly something that we want to make people aware of, because we talk with many people in our support community that receive a unknown variant, and then they're left wondering, right? Okay, well, when am I going to find something else out? And I have a cancer diagnosis, or other people in my family have a cancer diagnosis, and I am here, in the dark. So that's certainly something we're passionate about, and being able to help others that are looking for more answers.
Thanks for that, Carrie. And we share that passion. We want to pull information so we can better answer those questions. And we want to share back when we do learn that there's new information with participants. You sparked for me a question that I often get is, what is the timeline? for something, the variant of unknown significance, VUS uncertain, to be clearer. And the
Mhm.
really tough thing is there's no set timeline. I wish I could say, we'll figure it out in a year. Every variant will be reclassified, but it really varies. Some families will get more clarity in a month, a year. Others, it's 5 years, and we're still in that uncertain bucket. So we do what we can, and we want to pool our resources to try and help increase our understanding, but no set timeline, unfortunately.
So would there be a timeline you would suggest that patients should check back? Or is it more of a you'll get a call when there's a change and just kind of a wait and see?
Great question. So if we become aware of anything through our data sharing, we'll let people know about that when we become aware of it. But we also still encourage people to reach back out to their genetic counselors or doctor who ordered testing periodically, because we don't identify every single update through our data sharing. And it can be another nice way to ensure there aren't any other updates, or other updates that we might not share back with patients, like maybe there's updated testing they should have, or expanded testing,
That,
or maybe
yeah.
there's new guidelines for care that they need to be aware of. So my general rule of thumb from when I was in cancer genetics practice was telling patients every year or so, put me on your calendar, give me a call, reach out via the patient portal. It might be there's no news, but that'll be a nice prompt for us to get together and see, are there any updates for you?
And
So
it might be at that time that a genetic counselor might recommend additional testing as well, right?
yeah, depending on what testing someone
Available.
had, there may be more. Depends on their specific uh, situation, or might be an opportunity for the patient to update me on their family history. So it can be a
They
nice
learned
touch base.
something else. Yeah. Yeah. Wonderful. Well, we look forward to sharing this with more people. I thank you for all the work that you do. Um, and yeah, Nothing Pink is here to support individuals wanting to learn more about hereditary breast and GYN cancers. And we provide that personalized support across North and South Carolina. And happy to connect with you and let more people know about it.
Thanks so much, Carrie. We love all the work that you're doing. If anyone has questions after today, we welcome them, and happy to answer them.
Perfect. Okay.
All right. Thanks all. Have a good rest of your day.
Thanks, you too. Bye bye.